안녕하세요. 툴젠입니다. 2023년 툴젠오송 R&D 센터 기술 세미나를 시작하도록 하겠습니다. 첫 번째 순서는 툴젠 IP 사업본부 김유리 부사장님입니다. 박수로 환영해 주시기 바랍니다.
Hi, good afternoon. Thank you for attending 툴젠'S opening ceremony. My name is Yuri Kim, General Counsel and Korean and US Patent Attorney of 툴젠. 툴젠 is founded by the renowned scientist Jin-Soo Kim. So, let me start with this Nobel Prize story. So these two star scientists published a science paper in 2012, which the paper demystified how CRISPR-Cas9 bacterial immune system works in vitro. So after 8 years since this paper publication, and Nobel Prize came in 2020, which is 8 years after the CRISPR-Cas9 system is confirmed as a gene editing tool in eukaryotes. So somebody may wonder, why patent big fights in the US? So don't the Nobel Prize winners take it all? But no, here the winner does not take it all. There are many other parties. So one of the parties is Broad Institute. They managed to secure CRISPR-Cas9 foundational patent through a fast-track examination, earlier than the Nobel Prize group, aka CVC. Yeah, I will refer to the Nobel Prize group as CVC going forward. So obviously, the CVC group probably got furious, right? So they urged the US Patent Office to declare an interference against the Broad Institute. So what's interference? The interference is a special patent examination procedure. So actually, interference is not a patent litigation or anything, just a special procedure when multiple parties pursue a patent on the same invention. So at this point, we encounter an interesting question. So when multiple parties pursue patent on the same invention, and who would win? So there are two solutions, two answers to this question. So first, to file, and versus the first to invent. So the first to file system means so the government doesn't care whoever comes with this invention first. Just raise your hand first, then you will get the patent. This is very easy and convenient for the government, right? On the other hand, under the first to invent system, the inventor invented the invention first would get the patent, which sounds more fair and just, right? But it would rather difficult to find out who is the first, so that the patent law becomes more complicated. So now, every country on the globe adopts the first to file system. But up until 10 years ago, the US was the only country that adopted the first to invent system. So under the first invent system, if we take a look at the diagram, A invented the invention earlier than B, but B raised her hand first, earlier than A, right? But under the first invent system, still, A will get the patent because A invented first, right? But exactly the opposite under the first to file system. So because the CRISPR inventions, they were made before 2013, right? Before US switched from the first to invent to first file. So parties are fighting for patent through a special patent examination procedure, what's called interference, under the first to invent system, right? So the interference is a legacy of the first to invent system. So who are the participants? Who are the stakeholders of this patent dispute, patent fight? CVC or Broad, is that all? No, there is ToolGen, a Korean company. So 3 parties are fighting for the patent in the US under the first to invent a system. So if you take a look at this slide, between 3 parties, especially between the CVC and Broad, there are 2 arrows. That means this fight, they're fighting twice, right? So their first fight started in 2016 and it ended 2018, right? So the current fight, the ongoing fight, is their second fight. So in the first fight, the first interference between CVC and Broad, CVC, as I said, they urged that the patent office declared an interferences and the patent office accepted, right? So the procedure began. But at that time, CVC was with a CRISPR-Cas9 system only patent. The CRISPR-Cas9 system only invention. But on the other hand, the Broad group, their patents were about the invention of CRISPR-Cas9 use in eukaryotes. So if you look at the diagram and then on the Broad side, we can see the DNA, right? Eukaryote DNAs. But the patent office, the US patent office, they said, oh, there's no competition for the same invention. They are separate inventions, so we don't have to have We don't have to have the interference to continue. We can stop here. There's no interference. CVC will go with your invention, and Broad goes with your separate invention as well. So in a way, CVC failed, right? Their goal must be, taking Broad invention, right? So this is, of course, CVC appealed, but the Federal Circuit, the higher court, agreed with the U.S. Patent Office. This is an excerpt of the the Federal Circuit's decision. If we take a look at this, and then the judges cited actually the conversations between Dr. Daudna of the CVC group and her colleagues. See, a colleague contacted Dr. Daudna to inform her of Broad's success. Then he stated, "I hope you are sitting down. CRISPR is turning out to be absolutely spectacular in George Church's hands." What does it mean? It means, according to the judges of the Federal Circuit, it's applying prokaryotic systems in eukaryotes was unpredictable. It's just separate. It's not obvious. So they are separate inventions. But CVC did not give up. Less than 1 year after their defeat, they came up with another, interference. In their second fight, the CVC came with the same invention, exactly the same invention, CRISPR-Cas9 used in eukaryotes. But again, early last year, the Patent Office again raised Broad's hands. That means the Patent Office decided that Broad is earlier than the CVC in inventing CRISPR-Cas9 use in eukaryotes. Of course, this is very important foundational CRISPR patent, right? So they appealed. So the case is on appeal. So we are waiting for the Federal Circuit decision. So let's take a look at this slide. That means ToolGen is the earliest filer in the world of patent application which contains CRISPR-Cas9 used in eukaryotes. If you look at the slide, there are two CVC's applications earlier than ToolGen, right? But the USPTO says none of CVC's earlier applications contain any working example of CRISPR-Cas9 working in eukaryotes. That means there's no experiment data. So they cannot support the current, the patent, the current invention in dispute, which is the CRISPR-Cas9 use in eukaryotes. So ToolGen'S patent application is the first one in the world which contains working example, experiment data of successful data of CRISPR-Cas9 working in eukaryotes. So, ToolGen, according to the interference legal term, ToolGen is the senior party against CVC and Broad. As I said, ToolGen is the earliest filer, and so the ToolGen is the senior, and the other two, Broad and CVC. CVC later patent application contains experiment data of eukaryotes, right? So in this structure, ToolGen is the senior. And what's the senior? What's the benefits of being a senior party? So statistics say the senior party's winning rate is 75%. I think this is very natural because, you know, the earlier the filing, the earlier the invention, right? So, and during the interference, junior party, they must go first. They have to submit their arguments and evidence without knowing what senior party has. Just like shooting into the air, not at the target. By contrast, senior party can review what junior party says, and then senior party can submit tailored arguments and evidence. Then I'd like to just briefly explain how to win the battle, how the PTO can decide the earlier, inventor. So there are two ways you can prove that you are the first. You can prove either you made a working example of your invention earlier than your opponent, or even if you made your invention later, if you conceived the invention earlier and work very hard until finally you successfully made your invention, right? So that's the procedure. So, but I'd like to just emphasize that the conception is not an imagination. So conception, if I imagine a flying car, it doesn't make me an inventor. So the conception is not just an imagination, it's a definite and permanent idea of a complete and operative. I would like to emphasize the word operative, the working invention. So this is CBC and Broad's arguments. When is their conception date? When is their RTP? That means reduction to practice. That means they successfully implemented their invention dates. So they argued several conception dates and several RTP dates. Both parties argues that kind of dates, but Broad won. And as I said, the CBC failed. I would like you to get the hang of the procedure, how to decide which is the first. This is the evidence that the CBC submitted as to support their conception date. This is one of the scientists, Dr. Daudna's group's lab note. Dr. Inek's notebook, they argued, already they made their conception on March 2012, that this argument was not accepted by the PTO. PTO said that this is just a diagram. There's no concrete and there's no definite idea of how to make an operative invention. So this is not a proof of conception. Like this, any lab note, that's an RTP data. So that's the procedure. So let's take a look at interference timelines. See, ToolGen'S 2 interferences are on hold for now. until the appeal decision comes out, right? But CVC and Broad, they made the most of the extension of period, both parties. So the procedure is significantly delayed, almost 1 year kind of delay. So we originally we expected that the appeal decision would come out at the end of this year, but now that the first half of next year, probably the decision will come out. Then ToolGen'S interference will jumpstart right after the decision comes out. So, what if CVC fails to turn over the USPTO decision on appeal, right? So, many attorneys cautiously predict that Broad probably would win on appeal. If that's the case, and CVC will be removed from the scene forever. That means CVC will wiped out, and only ToolGen and Broad will remain and continue to fight. So I'd like to say, if somebody says either CVC or Broad goes to win, then it cannot be trustworthy. Here's why. Because nobody in the world but ToolGen knows what ToolGen has, right? But on the other hand, CVC and Broad, they already went through the PTO procedure and decisions. So their arguments and their evidence, they're out there in the public domain. But ToolGen'S story and ToolGen'S evidence, they're not. So only ToolGen knows what ToolGen has, right? So what if ToolGen wins? As a person, after I joined ToolGen in January 2021, more than 2 years, I have been scrutinizing CVC and Broad's evidence as well as ToolGen'S. So I'm confident that at the end of the day, ToolGen will emerge as the winner of the game. So what if ToolGen wins? ToolGen will be officially acknowledged as the US patent holder on CRISPR-Cas9 used in eukaryote patents. So all the companies using CRISPR-Cas9 in eukaryotes for the US market need a license from ToolGen. And finally, I'd like to just mention one ToolGen'S achievement as an example of ToolGen'S expertise in gene editing world. So ToolGen'S scientists, they are the first who published a paper in the world in which the CRISPR-Cas9 RNP transaction was successful, right? In animals and plants. Back then, DNA transfection is prevalent, right? So CRISPR-Cas9 RNP transfection is very challenging and was not so practical solution, but ToolGen scientists, they made it in animals and plants as well, both. So the papers ToolGen published are the first one in the world for CRISPR-Cas9 RNP transfection success. So even Nature reported the ToolGen'S success in CRISPR-Cas9 RNP transfection. And of course, much earlier than the paper publications, ToolGen filed and international applications for patent. So that's it. So thank you again. Thank you again, and I will hand over my mic to the second presenter. Thank you.
툴젠 특허에 대한 김유리 부사장님의 말씀을 들으니 툴젠의 앞날이 더욱더 기대가 되는 것 같습니다. 두 번째 세미나는 툴젠 치료제 개발실 이재영 이사님의 발표가 있겠습니다. 환영해 주시기 바랍니다.
Not too sure whether the slides are ready or not. Before I begin, I just wanted to apologize a little bit that I could not join for the morning session, so that I could not, I would have carried a lot of business card to exchange with you and to greet and meet with the people in both seed and therapeutic, and also some I saw some lawyers there as well. So because this ceremony meant to be a seed development's pretty much a glorious opening ceremony, so I don't want to overtake my presentation for the next speaker, which will be delivered by Dr. Han for the seed development. But I just wanted to say thank you all on behalf of therapeutic development at ToolGen for being here and participate in this technical seminar. So in terms of ToolGen'S application side, as Dr. Yuri just mentioned, that we do have a core IP, and there are two main pillars of the application side. One would be quite obviously the seed development, and the other one is the therapeutic development. And ToolGen Therapeutics have one specific goal, that is to translate this novel CRISPR-Cas9 technology to human therapeutics to help patients with incurable or intractable diseases. So we do. These are the therapeutic pipelines that are publicly available. Of course, I cannot really disclose the other therapeutic pipelines that we are incubating, but we do have both in vivo and ex vivo therapeutic pipeline. So in vivo means you directly administer CRISPR-Cas9 components in the body with the help of right delivery vehicle. And ex vivo means you take out the specific cells from the affected patients, and you gene edit or gene modify them and transfer back into the patient. For the in vivo program, we do have three main pillars which tackles the problems with the central nervous system or peripheral nervous system, eye and liver. And ex vivo programs involves the engineered immune cell therapy and also stem cell therapy. So the nervous system program involves TGT-001, which is the therapeutic candidate tackling Charcot-Marie-Tooth, which is often driven by genetic duplication problem in the peripheral nervous system. And also similarly, we do have a second therapeutic pipelines in the central nervous system, which is named Pelizaeus-Merzbacher disease, also driven by genetic duplication problem. And the reason why I highlighted these two in the nervous system program is because we are tackling the gene duplication diseases, which means that one specific good therapeutic strategy for the gene duplication can be applied to other indications with genetic duplications. And the main two problems that we are tackling is Charcot-Marie-Tooth 1A and Pelizaeus-Merzbacher disease. And our eye program involves the wet AMD. Wet means there is a vascularization problem in the eye, that is why it's named it as a wet AMD. And we're trying to tackle that with the important factor for neovascularization in the eye with the gene editing tool to tackle the problem with the age-associated macular degeneration. And our liver program involves two therapeutic candidates. One involves the liver biofactory platform, and the other one is the antithrombin platform, which I will discuss further later in the talk. And our cell therapy platform, like I said, involves the engineering the immune cell therapy, such as CAR-T or the engineered TCL-T. Or even the NK cell therapy, which we partnered with the Australian biotech Carterix. And also we do have an internal program involves mesenchymal stem cell engineering. And we do have an ongoing collaboration with the Korean biotech called CellArts-Gen in the context of limb ischemia. And just to give you a little further about the therapeutic pipeline, before I do that, I just wanted to let you know that the in vivo therapeutic strategy involves three main the R&D focus. One is to develop a novel gene editing therapy. We want it to be a little different from others in the field, like CRISPR Therapeutics or Editas. As we would like to basically develop a novel gene editing therapy to develop a first-in-class therapeutics to give maximal benefit or maximal value to the patients. And we also do have a disease modeling R&D focus where we do have a capability of mouse or rat gene editing to specifically model a in vivo model for the drug development. And also we do have a pluripotent stem cell gene editing so that we do have a basically an infrastructure in the therapeutic department to further expedite our drug development. And not to mention for the in vivo, like I said, we need a specific delivery vehicle to deliver into the body. And for that, we will need a very, very efficient delivery system. And we cannot do all this by ourselves. So we often do a open innovation or business development with the research and development, so that we're trying to maximize our efforts to deliver the CRISPR-Cas9 components to the specific the organ of interest. So to give you a little more details about our pipeline, so our lead asset is the TGT01, which tackles the problem with the Charcot-Marie-Tooth 1A, which is driven by the genetic mutation. Genetic mutation involves duplication in the peripheral nervous system, the genes. And because there is an increased gene expression level due to gene duplication, the affected patients often suffer from peripheral neuropathy, which is often involves limb deformity, muscle atrophy, and the patients will bound by the wheelchair at the end stage of their life. And cellular pathology involves damage to the neuron itself in the peripheral nervous system, and chronically damaged neuron will give rise to the muscle deformity and so on. The right therapeutic strategy would be, because there is a gene duplication, we would expect there is an increase in PMP, the gene expression level. And we try to tackle tackle this problem with the gene editing at the specific the regulator domain, which is basically the molecular switch, I'll say, to these affected genes, so that we're trying to dampen down the expression level to the normal level. And just to summarize some about the proof of concept data is that on the left-hand side, we have specifically identified the specificity of the CRISPR-Cas9 components, because imagine if you want to develop a drug development for with the CRISPR-Cas9, the off-target specificity will be the utmost importance. And we have utilized the 3 different methodology, which is in line with United States FDA guideline draft, and we have specifically identified that the specific CRISPR-Cas9 that we have developed for Charcot-Marie-Tooth have pretty much no off-target level. And in vitro-wise, the best possible in vitro cellular model would be the patient-derived cellular model, because it will harbor the same genetic mutation in the patient scenario. And with that, we have identified that when we treat it with the CRISPR-Cas9 components therapeutically, we have specifically knocked down to the normal level of this affected gene. And also in vivo proof of concept data using our TGT-001 showed that the CMT1A mouse will work better. And also there was an increase in the nerve conduction velocity, which is the electrophysiological manner. And most importantly, when we did a correlation analysis with the in vivo gene editing, basically if there's more gene editing in the in vivo, there's a significant improvement in the electrophysiology, which means that if there is a dose-dependent improvement in the phenotypic analysis. And the other program in the liver involves the liver biofactory platform, which basically means that we are trying to knock in a specific therapeutic gene of interest to the liver cells in the body, so that we're trying to make a stable, basically a factory in the body with the liver cells with CRISPR-Cas9 components. And with this, we can target quite a number of diseases involving hemophilia, where patients would have to do a repeated administration of the coagulation factors. And also, we can tackle lysosomal storage diseases, where patients have to undergo a recombinant protein addition therapy. And we are trying to develop this as the novel therapy, so that we discovered the gene called ApoC3, apolipoprotein C3, as a specific knock-in safe harbor site, which means that when you have a look at this transcript level, ApoC3 level is about 100 or 200-fold higher than the gene expression level that is associated with diseases that I mentioned before, hemophilia or lysosomal storage diseases. So which means that, as you probably know, the in vivo knock-in efficiency would be very, very low. Even if we do have a 0.1% knock-in efficiency, we can get about 20% of normal gene expression level, because we will be utilizing the ApoC3 endogenous promoter. And we did show a in vivo proof of concept data with the hemophilia B mice, which is published in Molecular Therapy last year. So that when we administer single administration of the CRISPR-Cas9 components, would correct the symptom of the hemophilia B. And also you can see the stable human factor IX expression level in the blood stream. Well, human factor IX is the gene that deficient in the hemophilia B. And also we also did a proof of concept data in the neonatal hemophilia B rat. Neonatal means that very at the very young stage, so that there is a active turnover of the liver cells. So active turnover means that the conventional AAV-mediated gene additional therapy would dilute out, so that the duration of the gene expression or the duration of the therapeutic efficacy won't last long. But then because we are knocking in a specific gene of interest to the liver cells, we did find that our liver biofactory is durable for quite a long time. We followed up for 6 months and so on. And we also did found a normalization of the hemophilia phenotype. And mind you, because the knock-in platform is, you can switch to the other, therapeutic protein or therapeutic gene of interest, so that we can insert enzyme, functional protein, antibody, or cytokines, and so on. So basically, we can do quite a number of things with one specific platform. The other liver program involves the antithrombin because tackling the hemophilia A and B. So hemophilia is pretty much driven by the deficiency in the coagulation factor 8 or factor 9, which is often involves this sort of a domino-like the molecular pathway of clotting. But at the end stage, we will need clotting for the hemophilia patients, because hemophilia, it means that there is no blood coagulation. And we're trying to bypass this strategy, and we're trying to tackle the antithrombin at the end stage of the fibrin clot to basically rebalance the hemophilia phenotype. And this is important because basically we can do a one-size-fits-all scenario, because the hemophilia patients would often undergo a repeated administration of the recombinant factor 8 or factor 9. So there will be some patients with these inhibitors, which is anti-factor 8 or anti-factor 9. So the patients won't be won't be able to take these available treatments. But if we're trying to tackle this antithrombin targeting therapy, we can do a basically one-size-fits-all. We can pretty much tackle all sorts of hemophilia patients. As a proof of concept, we delivered lipid nano CRISPR-Cas9 components to target antithrombin with the lipid nanoparticle, and we have achieved a very much stable and efficient gene editing frequency in the liver. And also we have shown a durable knockdown of antithrombin. Antithrombin is the secreted protein, both in the factor 8 deficient mouse model and also factor 9 deficient mouse model. And we have shown that there is a thrombin rebalance and in the both hemophilia B or hemophilia A mice. So basically, we have a proof of concept data for the one-size-fits-all scenario. And those were the in vivo scenario, and I would like to switch gear to the ex vivo scenario where we're trying to develop a novel cell therapy using CRISPR-Cas9 components. And there are two main focus of the cell type of interest. One is the engineered immune cell therapy. The other one is the mesenchymal stem cell therapy. And we don't want it to do all this by ourselves, because we know how critical and how difficult the CMC development for the cell therapy. So we will rather stay as a tool provider for the companies who are trying to develop cell therapies. And we have already established partnerships and collaboration with the multi-parties. And one specific example for the engineered T cell is the STIX-T platform, where we're trying to edit an important immune checkpoint molecule called DG kinase or DGK in an attempt to improve therapeutic efficacy against the solid tumor. And it would also we have also found that there is an increase in the durability when we edited DG kinase in the conventional CAR T. As you can see here, control. These are, mind you, these are second stimulated the tumor scenario in vivo. And as you can see, the conventional CAR T group, mind you, this CAR T is from basically similar structure to what Novartis has. And as you can see, there is a tumor growth in the conventional CAR T, whereas when we edited out the Dg kinase in this same CAR T, there is a specific tumor control. And with this specific data, we have established a collaboration with the Carterix. And I think you have heard a interview with the Carterix CEO, Ellen Tronson. And we have gone through a collaboration at the early stage in 2019. And we have licensed out our technology to them in 2021. And as you can see here on the bottom left, when they try to inject their conventional CAR T, non-gene edited CAR T, which is blue colored, there is a tumor rebound after a certain period of time. Whereas if we gene edit the Dg kinase in this similar CAR T, there is a superior tumor control in the T cell scenario. And also we have also further our collaboration to the iPS-derived NK, where we show that similar scenario to the CAR T, there is a superior tumor control. And very recently, Carterix actually sub-licensed the edited DGK edited the CAR T to the Chinese biotech Shunxi for the Greater China rights. So we're trying to do a clinical trials both in Australia and China at the moment. And yeah, so this is what I have prepared, and I wanted to thank you all again for the participation, and we'll try to make a further value with the therapeutic gene editing platform progress. And thank you all again.
유전자 교정 기술을 활용한 툴젠의 치료제 연구에 대한 기대를 높여주는 발표였던 것 같습니다. 그럼 마지막으로 툴젠의 종자 사업을 책임지고 있는 종자사업본부 한지학 전무님께서 발표를 진행해 주시겠습니다. 박수로 환영해 주시기 바랍니다.
퍼스널라이즈가 한다. Okay. Hello, everyone. Nice to meet you. My name is Jihar Khan. I'm in charge of the Seed Research and Business Development Division. I'd like to introduce the genome-edited seed development at ToolGen. Okay, before I get into the main issue today, I just wanted to share with you about the fact and expectations. Well, as you can see here, the 8 billion people and 60 billion livestock are living in this planet now. And then there will be 10 billion people and then 100 billion the livestock in year 2050. Okay. So we need to have more than 170% of our current crop productions to feed the human and the livestock. Okay. And unfortunately, we will face low yield than ever due to this temperature rise and also CO2 level increase. Okay, so the question is, what we have to do. Okay? What we need is new and the various genetic resources for the future food of humankind and the livestock. We should have crops with maximum yield, and then resistance to the climate change, and the resistance to the disease and pest. In order to fulfill the request, advanced breeding technologies are required. And of course, we can think about the other tools in order to improve the crop productivity. For example, like more fertilizers, more chemicals, like more arable lands by killing the old ecosystem. Okay, this is not the way that we can go. Today, I just wanted to focus on the breeding technologies. Okay, the human had domesticated, natural resources for a long time, about the 12,000 years, to get the edible crops, such as available germplasm. Okay, and then with those, the scientists have obtained the new germplasm, new lines, new cultivars. Okay, using the traditional breeding technologies for the last 100 years, plus by new breeding technologies. Okay. Now, this slide shows the milestone technology used for breeding practice for the last 100 years. As you can see here, hybrid, mutations, tissue culture, the GM technology, DNA mass is marker-assisted selections, and then genome editing, which is just a start. Okay, as far as I know, most of the major seed companies use all these breeding technology to get a single elite variety. Okay, so excuse me, without breeding technology, you can produce crops as much as what we have now. Okay. Now, the question raised is, what should a small biotech company like a ToolGen do to catch up or contribute to the global missions, which is developing crops for sustainable environment and high productivity and the resistance to the germs and bugs. Okay? We established the Seed Research Institute in October 2018, and then it became the Seed R&D division in January 2020. And then 3 months ago, this division moved down to this Osong Center, which is this building. Okay, the total area is about 20,000 square meter. You've probably already seen these pictures or numbers this morning, of which 5,000 is building area with the lab, so office and chambers, and then a greenhouse and so on. Okay, this summarizes what we're doing for the proposed and then activities. So we are developing the high value added crop using genome editing technologies, and then we will do seed sales in the future. So in order to achieve these proposed, we have done several activities for the last several years. Number one is R&D projects. So we're working on the R&D projects focusing on the climate change adaptations, functional trait, and disease tolerance, and so on. Okay. We also established the subsidiary in Kyrgyzstan in Central Asia, where we cultivate and harvest to make some revenue, although it is very small amount. And then also we're working with people in universities and companies and national institutes in Korea. And then also we are looking for the partners, okay, and overseas and for the R&D collaboration and then business as well. And then we also applied the genome-edited crops that we developed to USDA APHIS for the GMO regulation exemptions. And I'm going to talk about this later. And then also, we are working on the customer service. This is based on the consignment work, so I'm going to talk about this later too. Now, this shows the organization of SR&BD division, and the Seed Research 1 team is working with the potato, soybean, tomato, and rapeseed, the lettuce. And the Seed Research 2 team is working with Chinese cabbage and tobacco, corn, pepper. And we also have a seed management and the nurture team, and also seed business team. And as I mentioned that we also have a subsidiary in Kyrgyzstan, where we are making a small number of revenue now. Because we're selling the just ordinary soybean there, okay, not genomated soybean. Now, this is the map showing the location of R&D center and the subsidiary. And then Osong is here, as you can see, which is the 150 kilometers south of Seoul. And then here in Kyrgyzstan, we cultivate the soybean in northern part and also southern part, depending on the crops. Okay. Now, it shows the R&D project pipelines that we design. Okay. The projects that we're working on is located here, the left-hand side over there. And then, for example, the high oleic soybean and browning suppressed potato, herbicide tolerance corn, and also soybean, and acrylamide suppressed potato, and then solanine suppressed potato, and amino acid essential. I should say this way: the essential amino acid recomposition soybean, and drought tolerant pepper, and toxin suppressed tobacco, and soft rot tolerance Chinese cabbage, and so on. So once you develop this genome-edited crops, then you actually undergo phase 1 and 2 and 3 before the commercializations. Okay, so the entire process will take about 6 or 7 years depending on the crop. Okay, so as you can see here, what project has been done and then how much further each project has to go. Okay. Now, this is the research result that I wanted to share with you. And the first one is high oleic acid soybean. So we have developed the soybean that has the, the 80% of oleic acid in soybean seed, okay? And in the control group comprises about 20 to 25% oleic acid in soybean seed, okay? The second one is the browning suppressed potato, okay? When you skin, I'm sorry, I should say it this way. When you fill up the potato skin and then leave it on the room temperature for 24 hours, then it will become browning. As you can see then the left-hand side of the slide, right? And then oxidized, okay? But the genome-edited in a potato did not change the color, okay? And also we have successfully obtained the genome-edited corn and genome-edited soybean for herbicide tolerance. And also we have genome-edited pepper for drought tolerance. Also we have genome-edited tobacco for toxin suppressions. Okay. Now I just wanted to share about technical advancements that we have achieved while the other groups is kind of struggling to get this. And number one is transfection of RNP to protoplast. Okay, so after transfections, you can do the protoplast culture, and then you can go through the organogenesis, and then you can get the regenerated plants, and then you end up with the edited cultivars. So by doing this, comparing to the agrobacterium mediated transformations, okay, you can avoid the any DNA or gene contaminations to the host genome, right? By the way, we have a patent on this technology. Okay, second one is transfection to microspore, okay? Many times the scientists have trouble or experience that it's hard to get regenerative plants from the organogenesis, okay? While the, we can get the regenerative plants from the microspore through the embryogenesis, okay? So doing this way, we can have higher contents, higher probability of getting the regenerative plants, okay, compared to the organogenesis, okay? We've done this work, but the problem is the transfection rate is very, very low. Now, we also have a glycosylation modified crops, such as tobacco and lettuce for the protein productions, okay, by knocking out the plant's unique sugar enzyme, such as fucosyltransferase or gyrotransferase. Okay, so that we can avoid the unwanted glycosylations that might occur when we produce the protein or antibodies, okay, that are not originated from the plants. All right, and also we have the GM pepper, and then also GM Chinese cabbage that overexpress the Cas9 proteins. So we could induce the genome editing, okay, by inoculating the viral vector that has a specific guide RNA for the gene targeting, okay. So once we edited the GM pepper or GM Chinese cabbage, then we can have a seed from them, and then we can plant those seeds to the outside, and then we can get the phenotype, right? So we can take a look at the what the phenotype look like, and then this is a very good way to learn gene functions. So what we're doing with these seeds is that we are probably going to sell the seed and then license, or we could do the collaboration with other people, especially for the R&D. Now, we also applied the regulatory exemptions to USDA APIs, okay, with the genome-edited plants that we have, okay. And then USDA APIs approved the GMO regulatory exemptions to the petunia and soybean and potatoes, okay. And then recently we also applied another soybean, okay. So we're waiting for the review. Right? Now this is the customer service matter, okay. The seed business team is doing the customer service in order to get some revenue, and they're doing the synthesis of a guide RNA and they supply the Cas9 protein, and also they do the guide RNA validations using the protoplast, and then they also do the NGS service. And they do also have launched a genome-edited plant development service, okay? So that we could provide the genome-edited T1 generation within 1.5 hours, 1.5 years. Okay. Okay. I'd like to close my talk by saying that the ToolGen'S agriculture biotechnology research has been for the last 5 years, and we are going to continuously work on in developing the new crops, new germplasm, and then new items for the food security of humans and the livestock, and then also the environmental sustainability. Thank you for your attention.
앞으로가 더욱 기대되는 툴젠의 종자 사업에 대한 발표였습니다. 그럼 이것으로 2023년 툴젠오송 R&D 센터 기술 세미나를 마치겠습니다. 청취해주신 여러분 감사합니다.